Hey there. Welcome back to the Futurists. I'm Rob Tercek. This week, my buddy Brett King is flying around the world like he usually does. Means I can't get him on a call for the moment because he's sitting somewhere in an airport lounge or in first class or wherever he flies. Anyhow, it's just me this week. Thrilled to be back. Every week we bring you another person who's thinking about the future, and this week we're bringing back somebody who was one of our very first guests on the show way back when we started this a couple hundred episodes ago. Andrew Hessel. Andrew, my old friend from Singularity University. How great to see you again. Thank you for coming back on The Futurist. It's good to see you. How have you been?
I've been great, Rob. I get to play and for most of my life and just think about the future. Like, it's a good job.
It's a good job. More people need to be thinking about the future. That's why Brett and I decided to make this show. I know the term futurist rubs some people the wrong way. We had Jeff Jarvis on, and he gave us a little lecture about how pretentious the word futurist is. It was actually a good bit of show. Actually, it's kind of entertaining to hear it. But our thought is we can reclaim that term because rather than it being kind of a pretentious label for a public speaker, we think more people need to think like futurists. We think that's actually a great habit of mind. People need to cultivate an awareness of the trends that are going to shape the future, and also the habit of thinking about how that might affect them or their company or their business or their nation or the way the governance works or something. There's lots of future implications. A lot of people don't take the time to consider what might occur. And in some respects, that old adage, if you're not planning or shaping the future, then you're going to wake up one day and live in a future somebody else designed.
I don't disagree with that. And very few people think forward, particularly in the area of biology. We just take it for granted. Out of sight, out of mind.
It's great you bringing that up. So for the folks who don't know Andrew, let me give a brief intro. He and I met at Singularity University back when it was run at a NASA base in Silicon Valley, and we just clicked. This is ages ago, more than 10 years ago. But he was such a brilliant and fascinating conversationalist. Andrew's specialty is the field of synthetic biology. And we were just chatting a minute ago before the show started about how invisible biology tends to be. When we think about technology, we tend to think about computers and telecommunications. And lately, we talk an awful lot about artificial intelligence. It seems to come up like a bad penny every time we have this show. It's unavoidable. But biology is just something we take for granted. But Andrew, your perspective is that biology is, in some respects, it's the original technology and the most mysterious technology. Tell me what you mean by that.
Well, it's it's the original technology. It's the first technology on planet Earth. It made all the living things around us. But it's a mysterious technology because it's the only technology that humans didn't make. So, we've always learned to harness biology. It fulfills a lot of our needs. We're part of biology. But as we've, really developed science and engineering, we've explored it in a more systematic way, first by classifying, probing deeper with tools. Remember, when we made the telescope, turn it around, you've got a microscope. We started to realize the cellular nature of biology. And then we've really become better at dissecting it, really reverse engineering it all the way down to the molecular level and atomic level now. So it's a unique technology because, yeah, it's so mysterious. But as we've advanced it, we've gotten to the point where we've understood some of the ground rules, and now we're effectively beginning to engineer it.
This is an interesting long trajectory, right? So for the longest time, as you say, biology was a mystery, and frankly, life was and remains a mystery for many people. It's so profound a mystery that we have a lot of religious impulse and a lot of religious belief built up around it, around the sanctity of life and how to manage it and so forth. It really wasn't until just a few generations ago that we began to understand how it works and how cells divide and how traits are passed on from generation to generation. And even then, okay, we could observe, and sometimes we observed correctly, and sometimes we didn't. This is up to like the 20th century. I think Niels Bohr said biology with stamp collecting. Basically, you can collect samples of things and you can look at them and study them and appreciate them, but there's not a lot you could do to influence them, except for things like grafting, a kind of agriculture or agricultural techniques. Well, here we are in the 21st century, and now we're suddenly able to harness the power to rewrite biology. And that's really the core proposition of synthetic biology. And what makes it synthetic is the human element, right? That's the authorship part of it. That's where the humans decide what goes into a genome, what to take out. Can you tell us a little bit more about the process of synthetic biology for those who aren't familiar with it?
Well, in the 1970s, we learned how to do what was classically known as gene splicing or
Hmm.
recombinant DNA technology. And the way I like to describe it to people is that's learning how to write DNA code in much the same way that we would write a ransom note. You have to kind of start with, pre-synthesize, like
Yeah,
chunks of DNA, and you
yeah,
have molecular scissors and molecular glue, and you string it together. It always looks a little Frankensteinish, but you can write anything you want. It's just really tricky. And in my presentations, I actually have a slide where I took a quote from a synthetic biologist, Drew Endy, I sat down with a stack of magazines and scissors and glue, and I think it took me like an hour and 20 minutes to write out the quote. Now imagine trying to write out a string of genetic code, which is a molecule 2 nanometers wide, using molecular scissors and molecular glue. It's so hard, which is why, to really get started in genetic engineering back then required having the right lab, the right reagents, a PhD, lots of dedication. Then you could string together enough code to make maybe a protein. What's happened in my lifetime, and I still find it astounding, is that process of splicing, just like in splicing film and splicing music, has gone digital. So now we can sequence DNA with a machine called a sequencer, and which is effectively reading the molecule, the DNA molecule, and digitizing it. So now we can put it into an editor on a computer, and now we can edit it digitally like we do with other media. And when we're satisfied with our edits, we can send it to another machine called a DNA synthesizer, which is effectively just a laser printer for the DNA molecule. So impressively, genetic engineering can be done sitting down with a laptop now. And really, if you can type, you can be a genetic engineer. You're just not good at it yet. You don't be. The cool thing is, like, learning to program a computer. Once you learn the basics of programming DNA, there's only one language for
hmm
pretty much all living things. So it really, we found that there's been a tremendous acceleration in biology as it's digitized, because everyone's kind of working with the same medium. They're all trying to solve the same problems. The language is the same. And so even though it's gotten off to a slower start, in some ways it's a lot more unified than the world of computing.
Let's pause for just a second here because when you speak about this, you're using analogies and metaphors. You're kind of making a parallel to computer programming. But now I want to be clear for the folks who are listening. This is not exactly like computer programming. So, computers are digital technology. They use electronics. They record on magnetic media and so forth. But with a computer, you can make a digital representation of a real thing. We do that all the time. It's called modeling, and we can do that with an economy. It's when you do a spreadsheet, you're doing an economic model. Social scientists use computers to do models of society and social behavior and so forth. So we have models for everything, and gradually, computational science is transforming all the hard sciences. So we now have computational physics, and of course, computational biology, and so on. So that concept, I think most people are familiar with. Essentially, it's like the idea of applying software to almost any field, including science. But that's not what we're talking about here. Here, we're talking about is by analogy when we say programming biology or writing code and so forth. Those are analogies. And what you're really talking about are biological parts. You're talking about biological material, not digital material, not something that's recorded in electronic media or magnetic media. We're talking about things, wet things. And when you talk about writing, let's start with the sequencer. When a sequencer is functioning, what is it doing? Is it optically looking at DNA? Is it using chemicals to break down DNA and break it into its constituent parts? Is it a wet process? Is it a dry process? Is it using optical? Like, what is the method used to first read the genome sequence?
So I don't want to get into the guts of sequencing, but the process is important to understand. The DNA in cells is the operating system for the cell. It contains instructions to operate a living cell and to duplicate it. It's a pretty impressive program.
Yeah.
You have to think of it as the operating system. But instead of being constructed from digital bits, zeros and ones, the code is assembled as a long molecular string with the bits being chemical bits. And there's four of them that we abbreviate as A, T, G, and C. But it's a digital code. It's just not binary. There's four bits.
So it's
And the
quadratic.
order of that code literally instructs the cellular mechanisms to do things. There's machinery to read that molecule and act on those instructions. We know the lexicon, the order of the bits of DNA, the bases we call them, corresponds to the amino acids of proteins that ultimately build are the structural medium and the operational medium of cells. So even there are really interesting analogies between computers and cells. You can a computer is a digital processor. You can think of a living cell as a molecular processor, and you can translate between them. So a DNA sequencer will, when we get an organism, we can extract the DNA precisely, and the sequencer literally digitizes the order of bases in that molecular string and converts them to the digital letters ATGC on our computer. So it's really turning a molecule into bits, atoms into bits, which is great. And
Yeah.
then of course, once it's in our computer, we can manipulate it, edit it, delete it, shuffle around. It's easy.
Right.
Yeah. And there's certain rules for how that we've learned that DNA has to conform to.
And
And going the other way with
yeah.
the synthesizer, it's like a printer. We have the digital code on our computer. We send it to a machine that assembles the DNA molecule, and that effectively compiles the program that can go back into a biological system.
And again, the synthesizer is taking instructions, digital instructions, and they're turned into basically a recipe, if you will. Maybe that's a stretch, but it's turned into instructions for how to assemble amino acids in the right
No, you
order.
assemble DNA, you assemble the DNA molecule, and once you put that into the cell, the cellular machinery can then go and assemble the amino acids that build the
And then
protein.
it can reproduce, and that cell is you've effectively reprogrammed that cell to be a little factory that cranks out more of the same.
Yeah,
You
yeah.
know,
So DNA is essentially writing software for a biological system. And that can be a cell. And we even have what we call cell-free systems now, where we can take some of the components of the cell and put them in a tube, enough components so that when we put the DNA in the tube, it'll make a protein. It won't make a living organism, but it has all the machinery necessary to turn DNA into RNA and then into a protein all in a tube. So,
that sounds like that could be a valuable process, but people listening are gonna be saying, "So, what's it good for? Where would we need proteins?" Maybe you can answer that question. How would we use that to make novel proteins, and what would they be good for?
Well, proteins are the functional components of biology. So, they build the structure and they run the operational system. Proteins are some of the most magical biomolecules in existence. So, just as an example, for some proteins, we have to eat protein,
Yeah.
for our own building. So by engineering proteins, say a plant protein, we can actually make a foodstuff. Some proteins have functions. They can break down molecules or build up molecules. So today we engineer proteins that go into, for example, laundry detergents to break down the food stains on our clothes. Those are called proteases. They break down other proteins. I'm particularly interested in a type of protein called a polymerase, which is an enzyme, a piece of protein that can assemble other biological components. So the one I look at quite a bit is DNA polymerase. It's an enzyme that writes DNA. And then there's other proteins, for example, that we use for that we can produce in the tube, but then when we inject in us, stimulate our immune system to make antibodies. That's one of the ways we make vaccines. So there's many, biology is, when you think about it, a single cell, just one cell, if you can imagine it, we're made of 40 trillion, but just a single cell is kind of the most complex factory that in existence today. It's literally micron scale and can make thousands of different compounds, biochemicals, and regulate all these processes and even make more factories, all running, all dictated by a string of molecular DNA code. It's, to me, it's magical, but
Yeah.
we've reverse engineered it to the point where it's manipulated.
That's an astounding breakthrough. The generative capacity of nature is something that's almost impossible for humans to fathom in its entirety because it's so great. It encompasses us and all of our predecessors, but it's also the whole world around us. Just last month, I was inspired by a conversation we had with Kim Stanley Robinson, who spends a lot of time in the Sierra Nevada mountains. That's where he goes twice a year. He's been doing it for 50 years. And he often comes up with ideas for his books, his science fiction books there. And he inspired me to go to Yosemite. So I went there and I was in Yosemite. And as it turned out, I arrived with the rest of the group. So I was there for a couple of days by myself in the wilderness and laying in a tent, which is kind of fun. It's like this, less than a millimeter of nylon between you and the world outside. I was laying in that tent and I was thinking, like, wow, first, the scale is enormous, like all that teeming life around us. We don't tend to think about it because we're not in it that often, right? We're in a city, we're in a house, we're in a car, like we're in something that a human built, but it's very rare for us to be out there and exposed to nature. And even then, we're in a sleeping bag or a tent or something. But the scale was immense. And then the eternal nature of it, that this powerful force of nature is going to continue to exist for eons, long after we're gone, long after something else has replaced us, it will continue. So the prospect of humans being able to maybe assume control of that, or even exercise some measure of control over it and direct it, that's an awesome prospect. And it's
Yeah,
also
it's
awesomely
huge.
scary.
It's huge. I think it's the most important technology, and our our understanding of it and the beginning of our efforts to to guide it with intention is huge. I don't, people say, oh, you're playing God. And in some, well, we're definitely playing with the natural systems of this world because they're universal, and it has huge implications, and it demands responsibility.
Yeah, there's no doubt about that. When we first met, you made a good point to me, which is that the entire surface of the world is now designed. That we spend all of our lives in designed environments. And even, those listening might say, well, hang on, you just said you were in Yosemite, Rob. It's true. I was in Yosemite, I was in a national park, but even that is designed to the extent that we as a people agreed to set this land aside and leave it wilderness. But that was a decision that we made. And then, of course, there's the Park Service that is assiduously working every single day to maintain that wilderness. So it's not a completely wild space. There's very few wild spaces left on the planet. Most of the world has been designed by humans. And as a result, we have this, maybe we've sleepwalked into this responsibility. Maybe we're finally waking up to the fact that now we're the custodians of this planet. Now that we've designed it the way we want it to be, we've designed the food supply and the ecosystem, and all the natural resources have been harnessed towards whatever ends we desire. Suddenly we realize, wow, that means we have the responsibility. You break it, you bought it, right? We now have to manage this. And honestly, we haven't done such a great job of that. I'm certain that there are people listening to this, Andrew, who are going to say, wait a minute, we can't even manage fossil fuel. Why on earth would we allow humans to go monkey around with DNA and genome sequencing and genome synthesis and start to make new novel life forms when we have no idea what we're doing? How do you respond to that kind of statement?
Well, I look at it this way. Humans have become the most successful animal on this planet. Like, and that's, we've reshaped the world because we developed technology. We have our incredible brains. We're the most social animal. We're incredibly cooperative, and we've learned to leverage the forces of physics, et cetera, to, to guide our hand. This is huge, but our success has been built on relatively simple, crude, toxic technologies. Like, they've been very, they're just blunt tools.
Mhm.
Oil, burning things, mechanical processes. These are really blunt tools, but they're the foundational forces of matter. When you start going into the living world, you realize this is the most advanced form of manufacturing ever. Like a single cell, again, microscopic, um, can take elemental materials, carbon, hydrogen, nitrogen, oxygen, and transform them at standard temperatures and pressures that support life, and in some pretty extreme environments sometimes, whether it's high pressures, high temperatures, low temperatures in the Arctic, et cetera, and turn it into the most incredible molecular machinery. So it's like going from, bashing rocks with a hammer to suddenly having the most advanced, you know, uh,
Scalpel.
digital factories. Yeah, like it's
Yeah. Yeah, it's
incredible.
like a
So,
precision
so
instrument. Okay, that's a
so
nice way to put
for
it.
me, I find it inspiring that we've gained access to these tools, and it's going to reshape our world. Everything we make, everything we do, our health and medicine, et cetera. But we have to accept that we're beginners. And thankfully our AI tools, our digital tools, are going to help us along the way.
You know, it comes up in every episode. Artificial intelligence. You had to go there. Okay,
I
great. We're
had
going
to,
to take a little break.
but there's a
We'll come.
reason
We'll,
why I had to go there, Rob.
we'll
Like,
come back after the break and we'll talk about AI. And maybe we'll build on this analogy to computing because it seems like it's unavoidable in this context. Folks, you're listening to The Futurists with my guest, Andrew Hessel, the synthetic biologist, and we will be back in just a moment after a word from our sponsors. Hey, you're listening to The Futurists, and I'm Rob Tercek. And this week I am talking to my friend Andrew Hessel, the synthetic biologist. And Andrew's just been laying down the foundational concepts for what synthetic biology is all about. Now, much of what we've been talking about, Andrew, goes back to the Human Genome Project, right? This is kind of a big turning point where we went from maybe observing or, to use the snide term, stamp collecting, to actually being able to read DNA. And it was just, it didn't take a huge leap of imagination to say, gosh, if we can read it, maybe we can write it. Can you talk a little bit about that sequence of events? First, the Human Genome Project, and then the idea of writing the genome or rewriting it, if you will.
sure. The Human Genome Project, this idea that we should read our own code to understand how we're constructed and how we operate in
A
both.
lot of people thought it would be impossible. Like they were that as a vainglorious quest. It will never be accomplished.
Well, when it was proposed in the early 1980s, it was such a huge leap because at that time, the only thing we'd ever sequenced was short strings of DNA. I think the only organism we'd ever sequenced was a small virus, a few thousand bits of genetic code. And so to propose that, they had a pretty good idea of the size of the human genome, around 3 billion bits. That was a huge leap. but what they recognized, what this, this would be something that people could get behind, invest in, and it would be the tide that raises all ships. So it was launched in 1990. It was a 15-year plan, a budget of about $1 a base, so about $3 billion. And it was the biggest science project ever done in biology. So it just pulled in people from around the world. And for the first 5 years, most of what they did was break apart our genome and kind of package it in mini chromosomes so that they could start to organize and sequence the bits. And I think by 1995 they had about 1% of the human genome sequenced. But another group of folks were building better sequencers.
Yeah.
And in 1995, kind of the first fully automated high throughput sequencers were starting to come online. These were a game changer. And so, the whole between 1995 and 2000, it just accelerated like crazy. It was this massive race. And we went from 1% sequence, literally to over 90% sequence, what we called the draft in 2000. And really for the next, it almost took 20 years to close all the gaps in that draft. But we called it done in 2003 for the most part. There were some elements in our genome that are highly repetitive, and they were really hard to read with the technology of the day, but we've closed all that since. So yeah, we can now read genomes faster, better, cheaper. It was one of the most fastest evolving technologies on the back of the human genome, which I just call a $3 billion Kickstarter. And today we can do a human genome. I think the record is 5 hours to be sequenced and analyzed, which is pretty amazing. And it's a record that's bound to be broken. What I got interested in, though, was the other side of the coin very quickly, because I'm a bottom-up guy. I don't work with the human genome. I work with microbial genomes. And the first microbial genome was sequenced in 95. That to me was like, yay! And we've just, we've been adding them faster and cheaper ever since. But I also recognize what you can read, you should be able to write. And so I've tracked DNA writing technologies for a long time, and frankly, they're amazing. Today, if we can't write a lot of DNA in terms of the size of the strand, the string of bits. Most groups have optimized at under 10,000 bits of code, but that's enough to do protein engineering at scale, and there is a huge industry building up around exploring that space in medicine and structural materials and engineered enzymes, et cetera, et cetera, et cetera. I'm personally interested in, can we write entire genomes, like the entire code of the organism? And there's a reason why. Right now, we take an organism, we extract its DNA, and we end up with a file on a computer. And if we're really sophisticated, we end up with some sort of model of that organism on computer informed by the code. That's still a pretty big step from where we are for the most part, because we don't have a full dynamic model of even the simplest bacteria. So hold on that point. But I'm interested in how we go from that digital genome file back to the molecular DNA of the genome and boot it up to make the organism again. And we've demonstrated that we can do that for viruses. That was done back in 2002. And a team of scientists led by a doctor by the name of Craig Venter did that for the first microbe back in 2010. So, why aren't we printing microbes or teleporting microbes or doing all the incredible microbe engineering today? It's just the fact that the DNA writing technology, the other side of the coin from sequencing, has yet to really have that same super exponential advance that we saw with DNA sequencing. So that's where I put a lot of energy today. How do we make next generation DNA synthesizer assemblers?
And why is that difficult? What's hard about optimizing the synthesizer or writing longer bits of code?
Oh man, like the technology that has gone into DNA sequencing is astounding. You can go super deep on that, but it's harder with synthesis because most of the early DNA synthesis technologies used a chemical process. And,
Yep.
and don't get me wrong, I like chemistry, but when I did organic chemistry, you were really lucky if you've got 95% purity in your product.
Right.
Uh,
Right.
and because we've optimized the chemistry for assembling DNA, we've gotten up to over 99%. But it's a sequential process. You're adding a chemical bit to a growing strand, and if you're 99% efficient, you get to be 100 base pairs of genetic code. There's probably an error in there somewhere. Now compare that to the biochemical processes that our cells use to copy its genome. And remember, if a human cell divides, it has to write a human genome, right? Like it should be obvious and intuitive. But if a cell divides in, say, 24 hours, it had to write a human genome in that 24 hours.
So
Same
the
thing.
human cells, the cells are outperforming anything humans are capable of by a
Exactly.
factor of a million.
Yeah,
That's
and
really
so
quite
and
extraordinary.
that's yeah, sorry to speak over you, but that's
Mm.
because biochemistry is magical compared to chemistry. Like it's truly magical. So people have started to harness the biochemistry to write DNA. That's called enzymatic DNA synthesis, and that's had huge advances. It's so much less toxic. You can do it
Mm.
on smaller scales. So we've it's been breaking a lot of the performance capabilities,
But
but
that's basically mimicking what the body already does, what the cells already do.
it's leveraging, it's harnessing it. Like
Mhm.
we're using the same biological machinery inside of our cells, but we're telling it what to do essentially, and that's huge.
Is that similar to CRISPR technology, which basically they observe the process for how DNA could get snipped apart, and they're like, oh, let's get that enzyme and reproduce it. We can use that as a tool, a precision tool for slicing up the DNA. I mean, is that, I don't know if I got that quite right, but
Well,
candidly,
no, you've kind of hit the nail on the head. We discovered a biological system operating in this case, microbial cells. It was acting as a microbial immune system. Very simple. But if viral, genetic information came into the cell needed some way to distinguish between its own DNA and viral DNA, and it developed this mechanism where it could identify it specifically by this string of code, and then cut it and thereby inactivating it. We've understood that process and we've harnessed it as a genetic editor. But
and
remember,
that's
that's a wet
that's
bench.
uh that's
Yeah,
CRISPR.
that's CRISPR. But that's just making a cut or a deletion or a recombination. But
It's basically scissors. I mean, we're back to
it's
ransom notes.
we're back to scissors, yeah, and they're better scissors, but it's just scissors. I prefer my genetic manipulations to be done digitally and synthesized with 100% precision, no off target.
Okay, so that's a good point, right? So we when we grew up, there was this term genetic engineering, which was a little grandiose, really, because there was very little engineering happening. It was basically, like you said, ransom notes. You would chop up different parts of a DNA sequence and then smash them together and find out what happens. I mean, candidly, it really was that bad. It wasn't very elegant and it wasn't very precise. But that's changing now because now we have a good understanding of what parts you can stitch together and how to stitch them together, how to combine them. Uh, I'm as I'm saying these words though, I'm thinking that there's somebody in the audience listening right now going, "Oh my God, does this sound like Frankenstein? You guys are monkeying around with the laws of nature. You're messing around with God's work. What are you going to unleash on humanity?" And to be candid, there is a risk here, right? Because the same tools that could be harnessed that could be used to harness the generative capability of nature and maybe direct life or design life in ways that are better for humanity, maybe better for sustainability in general for the whole ecosystem. Those same tools could be used by a malicious actor to do something incredibly destructive. Like you could make a infectious disease that targets people based on genome so that you could go after a certain group of people who had a particular genome type. I don't know if that's easily done, but theoretically, right, that's one of the potential risks here is a kind of a weapon of mass destruction. Talk a
So,
little bit
so
about the resistance to this idea or the fears that you encounter.
Yeah, no, and I really, I'm very open about this.
Mhm.
Look, all technologies uh, are dual use, and that's because they can be directed by good or bad human intentions, right?
Yeah.
Like I think that's just as a baseline. I don't want to say biology is particularly different than any other technology and how it can be applied. We've learned to understand these processes, and along the way, we've made some of the best medicines ever.
Yeah.
And frankly, I think we're making the world better when they're used properly. That being said, and the only I tend to really look at what we're doing in engineering biology as a lot like software engineering and using our computers as an example.
Mhm.
And perhaps the biggest difference is biology's been at this a lot longer than we have, and everything in biology has defense systems. They've all got immune systems. Even bacteria, that CRISPR is part of its defense systems because biology is always eat or be eaten, right? So
It's
it really
true.
comes down to that.
Yeah.
So, but when you inject human creativity into the process, we can make things that nature really doesn't. Like, for example, there's very few viruses that are highly infectious and highly pathogenic because they'd wipe out all the hosts that they
Yeah.
infect, right? But that's something that we can engineer in the lab and could release. And maybe it would only be one release and it would spread and do its thing and then die out. But the worst case scenario in that is huge,
Yeah.
right? And yet at the same time, those viruses are being engineered to be vaccines, they're being gene therapies, they're one of the most powerful tools we have to do this research. That's the good side. How do we the asymmetry between offense and defense in biology is huge, particularly with these viruses, which have the smallest genome. So they're the easiest to synthesize with our relatively new technology, right? So I believe we are in the riskiest point of bioengineering today because we have the tools to engineer with precision, small things like viruses. And as COVID taught us, we don't have the defenses and
Mhm.
and and we need to solve that, and it needs to be solved for humanity, not just one particular country, etc. And
Well,
so
like I said, we're the custodians, right? We have to manage this whole planet now, including the life forms, and our life form is one of them. As you were just talking about COVID, it reminded me that one of the reasons we're healthy and able to have this call today is because we all got vaccinated with an mRNA vaccine, which itself is a product of this very process, synthetic biology. So we have synthetic biology to thank for the fact that that scientists at Moderna and Pfizer were able to collapse the research and development cycle for a vaccine from 4 years down to a matter of months. And part of that was because Moderna had done so much work prior to the pandemic that they already had a process in place. So they just had to get the exact genome sequence. But they had the sequencing done in that of that COVID-19 in January of 2020. So by February, they were already working on a vaccine. This is an extraordinary achievement, and I think it goes unreported. I think not enough people. First of all, there's all the crazy nonsense about anti-vax, so there's a lot of disinformation and misunderstanding. I think it's sort of sad that part of it, that reaction. But then even worse is our failure to recognize what a novel and breakthrough achievement it was to be able to use synthetic biology to create a vaccine that saved probably millions of lives, maybe more, maybe tens of millions of lives. And certainly for a lot of people, protected them from ever getting this disease in a terrible way. Um, I think that's an astounding achievement. But given all the hue and drama and and noise around COVID-19 and mask mandates and all the rest, it got kind of got lost in the noise. It's unfortunate because I think that would have been a great victory for synthetic biology, great evidence. Because prior to that, actually, Andrew, there was a lot of question about whether synthetic biology would ever see the light of day, whether it ever actually end up in a useful product. But now that same methodology is going to be used for, for instance, to fight cancer. You mentioned vaccines a moment ago. I want to circle back to that because I think, when most people recoil when they hear about, sorry, viruses. I want to talk about viruses for a second. When we hear about viruses, most of the time we're like, oh, virus is a bad thing. The cold virus, gee, we don't seem to have much of a defense against it. This is a bad thing. But your perspective is different. Your perspective is that a virus is just the fastest and most efficient way to deliver a payload to through a biological pathway to a particular point in the body. So for you, it's like the delivery system. It's the DHL courier of the body. Talk a little bit about the prospect of re-engineering viruses for good.
Yeah, no, I think it's huge. Like, it is a delivery mechanism. What's delivered could be malicious or it could be tremendous. And they're really specific to certain cells. We live in an ocean of viruses, and only a handful of them cause us any disease. And the cold virus is basically considered a training mechanism for our immune system.
Hmm.
Um, but as we build them with precision, we're going to use them in really sophisticated ways as treatments, as again therapies, as ways to reprogram cells, um, and not just our own cells, plant and animal cells as well. So I think viruses have come loaded with history of disease, and we don't appreciate the benefits of it, like the fact that they're probably the IP packets of evolution in natural systems. But I think our understanding of viruses and virus-like particles is going to be much more sophisticated moving forward. But I also, and so I'm generally very optimistic, but I also say we have a virus problem right now because the field of synthetic biology is going to have a really tough time flourishing if we can't secure it from viruses, in the same way that our computer networks would not have proliferated if every time you plugged in a computer, it was completely infected. We
Hmm.
don't have the antivirus systems in place to deal with engineered viruses at scale uh,
Mm.
currently. And I think that's and I'm not seeing the appropriate level of investment in technology and political attention and global cooperation to make what I tend to call an internet of living things secure.
Well, that's a super interesting prospect, the Internet of Living Things. Yeah, the security aspects, yeah, they sort of speak for themselves. We can all think of our own horrible scenarios there, but I want to actually close on a positive note because I want to talk about the possibilities that this unlocks. And for the folks who are listening, if I were a young man today, if I were entering college today, I would be focused on biology because I think that's where we're going to see the greatest gains. The last 60 or 70 years has been defined by gains in computing technology. We're going to see even bigger gains, bigger productivity increases, bigger breakthroughs, bigger scientific progress, greater benefits for humanity in biology in the next 70 years. And it's because that we're at the very foothills of understanding how these technologies work. And as Andrew said, we're rapidly developing a tool set. There's a lot of room for improvement. Now, some scientists talk about fanciful scenarios. George Church at Harvard talks about bringing back dinosaurs, literally like Jurassic Park, that's something that you could achieve with synthetic biology. Others were talking about synthesizing things like fossil fuel, so we don't have to drill for oil, although that's a little bit questionable whether the cost of that makes any sense. Where do you see the possibilities? What gets you excited? What do you imagine the future will hold in synthetic biology?
I'm generally an optimist. We are going to have some chop, as we move forward, for sure. These are disruptive. These are powerful capabilities that are poorly understood because life is not a technology that's well understood by anyone yet. But we've been harnessing it for thousands of years to improve the human condition. And I think that accelerates dramatically. Like, we get to replace, as you said, kind of drilling biological goop out of the ground and refining it to make the products around us and the energy, and the liquid fuels. And remember, a lot of it goes into the plastics and other components in the world that we live in. We can turn that around and say, hey, look, we don't want to pull it out of the ground anymore. We can take that carbon, that structural material, out of the air. now CO2 becomes a feedstock for everything we want to manufacture, not just trees. That's huge. Plants.
Harvesting pollution and re-engineering it using a biological process to
Exactly.
create something
And
useful.
lowering CO2 concentration all the while. So if we turn off the taps of sucking banked carbon out of the ground and incentivize sucking it out of the air, we never have to drill a hole again, which is huge. So that I'm generally positive, even around climate change, as we start to biomanufacture at growing and growing scale. So, certainly medicine and health are going to see dramatic improvements. The old ways that we have of developing drugs right now is so archaic that it really doesn't fit well with this synthetic biology, which is precision, and we can take it to the individual. Like that is huge. And so, we're going to see dramatic shifts on medicine beginning to end. I tend
So,
to focus on
so
cancer because
yeah.
cancer is a rare disease of the individual.
Well, and
And
you actually developed a methodology, right, for basically reprogramming a virus to target fast replicating cells, right? You could actually use a virus efficiently to destroy cancer.
yeah, it's one of my favorite ways of attacking a cancer because you're actually hacking the cancer with an engineered virus to make more, to make it a factory for more cancer medicines. I love that approach. And we and other groups have been increasingly successful at it. We're still going through a really, really archaic drug development process there, though, because no two people have the same cancer, and yet the drug development system and the investment system behind that favors one size fits all medicine. But that'll
Oh,
shift. Yeah.
I didn't
So,
know that. Oh, that's interesting.
but
Oh, so you're swimming upstream there just in terms of the economic proposition, how the drug
yeah,
industry works.
it's old systems. Things take time to change, but they will change.
Mhm.
Another really fascinating area is just in the area of antibiotics. I recently met Stephanie Strafty, who's famous for saving her husband's life, who acquired a resistant superbug while on holidays, and she single-handedly brought phage therapy, the use of viruses that infect bacteria, to the West. It's been widely used in Georgia in overseas for as the standard of practice, using these viruses as antibiotics. But we had chemical antibiotics in the pharmaceutical industry, so it never really caught on here.
Did it cure her husband? Did she cure
She
his
did.
illness?
Yeah, no,
Well, that's really
and
cool
wrote
because
a
this is a
book
big
called,
issue, like
yeah, yeah.
resistant antibiotic resistant bacteria. That's a gigantic issue because of the overuse of antibiotics.
Yeah, I don't
Ooh.
I don't see that as being an issue going forward as we start to engineer antibiotics with precision, including these what they call phage-based antibiotics. Huge opportunities there for knocking out microbes.
And
She wrote a brilliant book called The Perfect Predator. I encourage you to check it out.
good. Okay, so we talked about energy. We talked about medicine and healthcare, antibiotics. But also, let's not forget, 25% of our economy is food.
Yeah.
And it's all biological, right? So the sourcing of that, are there applications for synthetic biology in food production?
Oh, we're seeing it, with the Impossible Burger. We're seeing with plant-based medicine. We're seeing it with culture, what they call, this cellular agriculture. Instead of growing a cow for meat, why not just grow the meat? We've seen it with dairy products because really that's just we can engineer cells to produce dairy proteins in the same way that we make beer, for example. So, a company.
That's literally like harnessing the cell as a miniature factory. And if you
Mhm.
put enough of those together, that miniature factory can start to crank out a product, a useful product. Like we talked about earlier, making proteins. You can actually make specific proteins that are food for humans or
Yep.
animals. Now, the other thing you mentioned is, and people tend to forget this, oil isn't just used to fuel our cars. It's also used to make stuff because plastic is a derivative. It's a petrochemical. Um, so talk about material science. I know at one point we spoke about, I don't know how serious this was, the idea that you could plant a seed for a tree and it would grow into an engineered house, into actually like something you could live in, like a biological house. Is that fanciful or is that possible?
Oh, it's fanciful today, but as we gain the ability to program entire genomes, and again, that synthesizer assembly technology today is not up to that task, which is why I push for us to get there. Today we can synthesize kilobits, the kilobases of DNA efficiently. I want us to get to the megabase scale and then to the gigabase scale sooner than later because that it just unlocks creativity. And when we get there, yes, we'll be able to make seeds that, we literally put in the ground and we will grow, living structures, that nature could never produce because there was no selective pressure to ever produce it. But the beautiful thing about engineering biology and doing it with our intention is we create the environments that these things will live in, and we can pick and choose from code that nature has generated across all the tree of life. So there's no species barrier when you're working with at the code level. So I love this. We literally cannot imagine the things we can create just with standard biology, the standard biological code, by learning to mix combinations and print them ourselves.
Super fascinating and inspiring. Andrew, it's always great fun to see you. Thank you so much for joining us on The Futurists. Tell me, for the folks who are listening that want to learn more, where do they find out about you? Where can they read your work or find your papers or your website?
I've got a book called The Genesis Machine that I wrote with Amy Webb. It came out a couple of years ago. That's a great one to start. These days, if you just start asking and looking at the term synthetic biology or digital biology, you'll find a ton of material, including compilations of some of the key papers, which is great. But I just want to steer you back, if I may, to one last thing, because I mentioned it, AI. Jensen
Yeah.
Huang, the CEO of NVIDIA, was asked about a year and a half ago when he gave a talk in Berkeley, what's the next big thing after AI? And he said, absolutely, it's digital biology. As
Mhm.
biology shifts from being science to engineering, it changes the world, which is huge. More than that, we're already training these AI systems to read the code of the language of biology, and it can read it natively. It can speak it fluently. So it's not going to be us that gets down and starts designing A's, T's, G's, and C's any more than we don't write computer code with zeros and ones. It's going to be these AI tools, and we're going to be prompting them and guiding them with our intentions it's transformative. So
So synthetic biology is machine readable at this point. I suppose
it's
that's the
all
whole
machine
point.
readable, and the computer tools are designing biology now. And literally, people have already shown examples where they've built these models and they've said, "Hey, let's make another version of this enzyme. For example, green fluorescent protein, which is an amazing enzyme that glows." But let's make it really different than the green fluorescent protein in nature, like by more than 10%. And they've just let the AI models kind of rerun evolution and come up with completely orthogonal proteins that do the same function. And
Hmm.
this is huge. It's huge in terms of intellectual property. It's huge in terms of designing new functions. It's just massive. So these AI tools are literally going to be the master genetic engineers that we just prompt towards the goals that we want. And it's happening so fast that it's astounding. So again, we can't translate the digital designs to biology, to reality fast enough because our synthesizer assemblers aren't great yet. But I expect that in the next decade, we are going to see huge jumps in the performance of DNA synthesizer and assemblers. And that opens up a bigger and bigger cone of possibility designing, the biological future.
Super. Well, thank you very much for joining us on The Futurists. Always a pleasure to see you. And folks, in this show, we'll be bringing you a different futurist every week, someone who is thinking about designing, planning, influencing the future in their own way. Next week, we'll be back with Brett King. He'll join us again, and we will have another guest for you. We're welcoming all kinds of comments on social media, any kind of suggestions that you have, we welcome that. And finally, I want to give a big shout out and thanks to our friends at Provoke Media who make the show possible. So it's always a pleasure to see you, Andrew. Great fun to do this show with you, and I will see you all in the future.
Thanks, Rob.